Okay, so good afternoon to everyone from Spain and good morning for the people in the Americas. It is my pleasure to introduce our next speaker, Francesco Volpe, a second-year Ph.D. student at the University of Turin in Italy, working under the supervision of Dr. Domenico Bosco. Originally from Terlizzi, a small town in the Apulia region, Francesco developed a deep interest in entomology and plant pathology after witnessing firsthand the impact of Ooctonus Vulgatis in his hometown. His academic journey into the world of Xylella fastidiosa began during his undergraduate studies, and it has since evolved into a focused research effort aimed at finding environmentally sustainable solutions to manage its primary vector, Philaenus spumarius. Francesco's current work explored the use of Ooctonus Vulgatis as a potential biological control agent through its parasitism of the insect eggs. He's also investigating the application of entomopathogenic fungi as an alternative strategy for the vector management. Francesco, whenever you are ready, please begin your presentation.
Hello to everyone. Thank you for introducing me. Thank you to Blanca for inviting me. I'm happy to be here to share with you the results about our research on Ooctonus Vulgatis. I would like start this presentation with a brief discussion of the title that I've chosen for this presentation, Between Promise and Reality, Evaluating Ooctonus Vulgatis as Biocontrol Candidate for Philaenus spumarius. Why Between Promise and Reality? This insect seems to be a promising biological control agent because is naturally present in Italy, but also in Europe, could reach a high parasitization rate, and it could work autonomously. I think that the last point could be interesting and important in the Apulian system, in which often the management of the vector that is crucial to contrast the spread of the bacterium has some obstacles related to the presence of some olive groves abandoned or uncultivated land, but also for the presence of country road with host plants in which is challenging the management. I think that a biological control program with an egg parasitoid could have an effect also in these areas. But the reality is harsh, and I will try to explain this in my presentation. Before talking about Ooctonus Vulgatis, put some context about natural enemies of Philaenus spumarius. In literature, there are few data available about natural enemies. There are some reports about spiders, ants, diptera, but also other more strange natural enemies like frogs or birds. There are some reports of episodic events caused by entomophthales fungi. And recently, some studies report the activities of some entomopathogenic fungi strains against Philaenus spumarius. Only two insects have been studied to be used as biological control agents of Philaenus spumarius. The Reduvide Xylellus renardi, that is a predator, but is generalist predator. And the predation in nature seems not to be an important source of mortality. The other one is Verralia aucta. Verralia aucta is a parasitoid of the adult stage of Philaenus that kills the host at the end of season. In this way, vectors can still spread the bacterium. Moreover, the parasitization rate of Verralia aucta is low, up to 5% in Apulia region and up to 17% in northwestern Italy. But this parasitization seems to be more higher in United Kingdom, up to 40%. In this context, the egg parasitoid could be promising biological control agents because they can reduce the vector abundance before its epidemiological role. They are able to kill insects at the egg stage. In literature, there are few egg parasitoids of Philaenus spumarius reported, 2 unnamed species of the genus Centrodora, and some reports about parasitoids of the genus Ooctonus. Ooctonus vulgatus is an egg parasitoid of the order Hymenoptera, of the family Mimaridae, that is naturally present in Europe and in the USA. There are about 200 occurrences in literature. You can see the reports in the map on the right. And the biology and ethology of this insect is really poorly known. In fact, there is only one paper in this moment in literature. This insect was reported for the first time as potential biocontrol agent of Philaenus spumarius in 2020 in Corsica, in a work published by Mesmen et al., in which 4 sites in Corsica have been monitored for the presence of this egg parasitoid. And Ooctonus emerged in 3 out of 4 sites with a parasitization rate up to 69%. In this work was produced also a prediction model. There is a map in the slide on the right for the presence of this insect in Europe. But there isn't a relationship between this presence and Philaenus spumarius, but this model is related to records of Ooctonus vulgatus in Europe. About the parasitization rate, that is high, we must say that in Corsica there is a peculiar situation in which Philaenus spumarius, that commonly, like other hafrophorid insects, lays on dead vegetables materials like straw, lays in the underside of the leaf of the plant Cistus mons beliensis. There is a strongly association between the insect Philaenus spumarius and this plant. We can see this association in this photo, in these pictures. There are the Cistus mons beliensis plants and eggs laid on the leaf. This photo are made by Mesmen, and also in these pictures you can see the high abundance of Philaenus spumarius on this plant. The question is, what is the situation in mainland? What is the situation in Italy? Further studies are needed to evaluate the possibility to use this insect in a biology control program. For this reason, the aims of this work were evaluate the presence and prevalence of Ooctonus vulgatus in northwestern Italy, and study its biology. Try to evaluate the parasitization rate of Philaenus spumarius. To do this, we have done a field survey in the last 3 years using the sentinel eggs technique to evaluate the presence and prevalence of this insect in northwestern Italy. We obtained egg masses of Philaenus spumarius and exposed them in 10 sites in the last 3 years, 5 in Piedmont and 5 in Liguria. During the period autumn-winter, that is the period in which the Philaenus spumarius eggs are naturally present in field. During the first year, 2022-2023, we exposed about 1,500 eggs. We obtained these eggs from mass rearing, in which there were insects sampled in Chieri, in province of Turin. We keep this insect in mass rearing for the oviposition using pieces of straw as substrate for oviposition. And in the end of October, we retrieved the substrate, counted the eggs, and we exposed them in 2 different setups. tent and cylinder. In this slide, there's pictures show the first setup, the tent, that is inspired to the way in which a German researcher, Werner Witzak, in 1973, exposed eggs spumarius in his trial to protect them from predator like bird. The second setup was the cylinder, a setup in which the eggs were in a pot with wet perlite and wet filter paper with the straw in which there were egg masses, protected by this metallic net. About the sites, we exposed in different sites like olive groves, fallows, vineyard, in which the abundance of the target of the Philaenus was known to the research group. We exposed for each site 3 tents and 3 cylinders with about 60 eggs each one from November to February. This slide shows the workflow. At the end of the exposure period, the eggs have been retrieved, counted, and placed in petri dishes with wet filter paper and honey drops, eventually for egg parasitoids. And then these petri dishes are kept in growing condition and checked every day until the emergencies. At the end of the first year, we retrieved the 60% of eggs, and 92 egg parasitoid emerged with a sex ratio female biased. All of them have been identified using the dichotomotus scale of triapizin as Ooctonus Vulgatis. There are 2 pictures on the right. And we have done also the molecular identification using COI gene that confirmed the morphological identification with a match of more than 99% with the sequence uploaded for Ooctonus Vulgatis by Mesmer. About the sites, 4 sites out of 5 have been parasitized with an average of 17%. During the first year. During the second year, we tried to improve our work, and we exposed 10,000 eggs of Philaenus in 3 different setups. We used the same 2 setups of the previous year, tent and cylinder, and we had one other setup, sentinel reading. In this setup, eggs were directly laid in field by insects, Philaenus encaged in this cylindric isolator, where there were plants for the maintenance of insects and straw for oviposition. There were 10 females and 5 male. About the time in which we had exposed the sentinel eggs in the first year, we probably missed a time window in which the eggs could be present in field. In the slide, you can see the period in which we exposed the sentinel eggs in yellow from November to the end of January. But the oviposition of Philaenus at our latitude start in September and end in December. For this reason, probably there is this missed time window, and to enlarge the window of exposure of sentinel eggs during the second year, we tried to force females to lay in August to have eggs ready to expose in September. We used a modified protocol for the parapause breakage published by Morente et al. In 2021, and we used these eggs in the first period from September to October, and we used eggs naturally obtained in reading in our facilities, like the previous year, in the second period from November to December. By the way, in each site, we exposed during the first period, forced eggs obtained by the parapause breakage protocol in 1/10 and 1 cylinder in each site, 100 eggs per setup. In the second period, we exposed 3/10 and 3 cylinder per each site from November to January. And we exposed the sentinel reading, the new setup, 2 per each site in both periods, from September to January. About the trial of the second year, we retrieved 8,000 eggs, and more than 600 Ooctonus Vulgatis emerged. Also in this year, the sex ratio was female biased, and most frequently parasitized setup was the sentinel reading. 7 out of 16 setups have been parasitized. In the graph on the right, you can see that only 1 cylinder in Liguria, only 1/10 have been parasitized in this year. About the parasitization rate, there was a very high variability in a range from 0.5 to 43%. The parasitization rate was higher in Liguria, and we can see these differences in the graph on the right, and probably these differences in the 2 regions could be related to the different climate conditions that could affect both the development of the eggs of the host, and in this way, the capability probably of the egg parasitoid to parasitize them, but also this could have an effect on the presence of the egg parasitoid. About the results, the performance of the new setup, we retrieved a high number of eggs laid in this setup, about 400 eggs in each setup. And in the picture, you can see the status of the isolator at the end of 4 months of exposure in field. Moreover, seems to be a correlation between the number of parasitoid that have been emerged in field and the number of eggs in the setup. But this data is preliminary and require further statistical analysis to be discussed. During the second year, we tried also to estimate the longevity of this insect that was 10 days, but Petri with 5 of 6 insects, I don't remember in this moment, survived for more than 2 months in a climatic chamber at 6 degrees. Moreover, in the 2 photos on the right, you can see some signs of the parasitization. The parasite's eggs become dark in the last part of development, and the parasitoid, when emerged, leaves a circular emerging goal in the egg masses. During this year, we used only the best setup of the previous year, the sentinel reading, and we exposed this setup 2 sentinel reading per site only in the period from September to December. About the results of the third year, we retrieved only 2,000 eggs, with a high proportion of broken eggs, probably predated, I don't know. This year, the status of eggs wasn't very good. At the end of the period in which the petri dish is with eggs, at the end of the period in growing condition, about 900 of Philaenus neonids emerged, and only 27 Ooctonus Vulgatis emerged. If we see the data about the parasitization rate in the graph on the right, we can see that the parasitization rate was extremely lower than the previous year. But this year we found some egg parasitoid dead in eggs in early January, in the first week of January, and we found also some signs of the parasitization, always early in January. Probably the parasitoid emerged earlier than the previous year. In these pictures, you can see the parasitoid that died death in the egg masses, but also the signs of the parasitization. These pictures have been made in the beginning of the second week of January. About the sites, only 3 out of 8 sites have been parasitized, and the eggs laid in sentinel reading was lower than 2024. Probably this could be related to an entomopathogenic epizootic event that we have found in the collecting site. We don't know, but probably the insect in the sentinel reading died before to complete the oviposition. About the parasitization rate in the 4 setup parasitized was low, but if we add the number of the signs and the egg and the egg parasitoid died in found died in death in the egg masses, the parasitization rate reach on average the same values of the previous year. About 10% in Piedmont and 50% in Liguria. During the second year, we tried also to do some biological trials in lab to evaluate to try to obtain a reliable parasitization rate of this insect. We offered 2 types of eggs to Ooctonus vulgatus, a female of Ooctonus vulgatus, that was kept for 24 hours with a male for the mating. We exposed fresh eggs and old eggs and used these eggs in 2 types of tests, double choice test and no-choice test. The fresh eggs, in according with the description of Witzak, were eggs in diapause, and old eggs were eggs with the embryo more developed, eggs quite ready for the hatching. At the end of these trials, parasitoid emerged in 1 replicate only of the double choice test, and only fresh eggs have been parasitized with a parasitization rate of 50%. The strange data is that the premaginal development duration of Ooctonus vulgatus in these eggs was about 2 months, and this data is strange. Why? I don't know. We have some hypotheses. Probably the timing of the exposure to Ooctonus vulgatus female was too long, or probably the egg stage was too developed. I think that probably the description that we have about the development of the embryo and the morphological description that we have of Philaenus spumarius eggs aren't enough to discriminate the different stage in the embryo development and the different stage in the diapause. To discriminate eggs to offer different egg to offer to the parasitoid. This year we had few samples to try to do the same work, and for this reason we tried to evaluate the oviposition strategy of Ooctonus vulgatus. To do this, 18 females have been dissected to count the number of developed eggs inside. We have done the dissection in two times. We dissected 2 groups of insects. A group of insects that was 1 day old, and one other group that was 7 day old. About the results, we can see that when the female of Ooctonus vulgatus have eggs developed in average 20 eggs, and we could say that Ooctonus vulgatus seems to be quasi-provigenic. But after 1 week, the eggs loaded in the ovarioles increases in a significantly way. I think that these data are very preliminary, but could be useful to understand the ethology of this insect. To discuss this data, what we know about Ooctonus vulgatus. Ooctonus vulgatus is present and widespread in northwestern Italy, but was found also in Apulia, in two sites in the north of Apulia and in the south in Salento. And I think that other records could improve the prediction model made by Mesman. The parasitation rate in field is variable and lower than in Corsica. I think that this could be related to the peculiar situation in Corsica. I think that the parasitoid could be more easier find eggs on the plants than in on the ground than at the ground level, and probably there are some tritrophic interaction between relationship between the egg parasitoid, the host Philaenus spumarius, and the plant. Moreover, the parasitization and the rearing in lab condition is not straightforward. We need other info to evaluate the possibility to use this insect in a conservative or augmentative approach. Arriving to the harsh reality, the study of this insect is challenging for a lot of reasons. For example, the oviposition behavior of Philaenus spumarius makes difficult offer fresh eggs to the parasitoid and require a long work. And also for the lab condition, and also in field, the exposure time should be shorter to understand when and what type of eggs are parasitized. Moreover, the Philaenus spumarius is a monovoltine insect, and this means that we must wait 1 year to obtain other data at the end of one trial. Reply to this question require a labor-intensive and long research. The second point, alternative hosts. We don't know where this insect goes in summer, if have and what are other hosts. But these information are very important, for example, to evaluate off-target effects, but also to obtain mass reading that is crucial to do a biological control program. In literature, Huber in 1986 reported other hosts for the genus Ooctonus. Among Cercopidae, there is Afrophora salatogensis, and among Cicadelidae, there is Nephotetix cincticeps and Cicadella viridis. The first two species, Afrophora and Nephotetix, aren't present in Europe, but Cicadella viridis is present. Cicadella viridis in warm condition could reach 3 generations per year, but I think that try to synchronize the cycle of Ooctonus vulgatus to the cycle of Cicadellas is challenging. And also for this aspect, the reply of this question require a labor-intensive and long research. Third point, Ooctonus vulgatus is native natural enemy, and probably its effect against the target in feed could be lower, but it's an insect already adapted to our environmental condition. To conclude, further investigation to assess the possibility to use Ooctonus vulgatus in a biological control program against Philaenus spumarius is needed, but we have to find effective ways to cope with these challenges. I'm ended. I want to thank you for your attention. I want to say a special thanks to my supervisor, Dr. Nicola Bodino, to my professor, Domenico Bosco, but also to all research group for the support in my work. Thank you.
Thank you very much, Francesco, for this interesting presentation. So now we will open the floor for discussion. So feel free to ask directly by unmuting yourself or type your question